Hello, welcome to the lecture, and I'm Thomas Ming-Shui Chang. I've been working in artificial cell for more than 60 years. It's a pleasure to briefly summarize this area for you. First, let's ask a question. This is an example of the possible uses. I'll let you look at it. Microdialyzer, drug delivery, blood substitute, biotherapeutic, vaccine, cancer therapy, immunotherapy, enzyme gene therapy, cell therapy, biotechnology, nanotechnology, regenerative medicine, agriculture industry, nanocomputer, nanobody, nanosensors. Let's look at enzyme gene therapy. Enzyme inside artificial cell doesn't come out, is excluded from immunological action outside. This solute substrate can go in, be converted to product, and come out. First report, use it for urea removal, use it in congenital hereditary diseases, use for cancer, use for bilirubin removal, and use for multi-enzyme with cofactor recycling. We can implant artificial cell into the body to work in next to biological normal body cell. For instance, we implant asparaginase artificial cell for lymphosarcoma, and the result shows that for those injected with control artificial cell with no enzyme, the number of animals having tumor continue to increase. In the case of those injected with artificial cell with enzyme, only 50% of the animal show the tumor. These type of injection for tumor cancer does not require repeated long-term injection. So as a result, these are now being used in the form of PEG by another group for routine clinical use. Another one is to inject artificial cell containing enzyme for hereditary enzyme defect. For instance, for academic mice, the elevated level is decreased by injection of artificial cell. In this case, hereditary enzyme defect require long-term treatment for the remainder of the life, so repeated injection of artificial cell will not be practicable, but this could be solved by solution is to use oral injection. For instance, after oral injection, it moves down intestinal tract and is then excreted after it completed its function. Oral administration. For instance, in Leschner disease, it's an enzyme defect that's very rare, and the Montreal Children's Hospital contact me if there's something we can do. The trouble is hyperxanthine increase, and we give artificial cell containing xanthine oxidase obtained from fermentation. This is converted into uric acid that is excreted. The result reported in Lancet show that it's effective in decreasing hypoxanthine after 1 week. However, this is a very rare disease, so we are more interested in looking into a more common one, and the most common one is phenylketonuria. In this case, phenylalanine increased to toxic level. So we give artificial cell containing fermentation obtained phenylalanine ammonia lyase. The phenylalanine is converted to cyanoic acid. This is converted by liver into hepatic acid and then excreted in the kidney. The result shows that normal animal has low phenylalanine level. PKU rat has very high level. Those on PKU diet has lower level. Those with oral artificial cell has level comparable to the normal rat. However, the enzyme is too costly. So as a result, we collaborate with IBEC company to produce recombinant enzyme is more practicable. And as a result of that, IBEC collaborate with a company in the US to prepare PEG cross-linked to the recombinant enzyme, and this has already been approved by FDA. Finally, may I invite you to visit the website. We don't have time to go into any detail. This is just a very brief summary. If you are interested, the detail in articles, videos, and books are available. If you have any question, I'll be happy to answer, and this is my email address.